Hello students, I am Puneet Bajpai and today I am going to discuss about a very important topic of class 11th NCERT, that is biomolecule. So let's come in our topic. So first of all we should know about about all the molecules that are present inside a cell. So we can say that cellular pool. A very important subtopic of biomolecule is cellular pool. So cellular pool is the is quantity of all the molecules that are present inside a living tissue. So we can say that inside a cellular pool there are two subcategories are divided, that are inorganic molecules and organic molecules. So all the molecules that are present inside a living body are called as cellular pool, and this cellular pool is subdivided into two important categories, that are inorganic molecules and organic molecules. So inorganic molecules are the different types of element, that is potassium, calcium and other type of minerals. And when we talk about the organic molecules, that are called as biomolecules. Okay, so this is the important definition of the biomolecule, that is all the organic molecule which are present inside a living tissue are called as biomolecule. So we should know about the definition of the biomolecule when we study about the biomolecule. So the definition of the biomolecule is that all the organic molecules that are present inside a living tissue are called as biomolecule. So this is the important definition. And now these biomolecules are subdivided into two categories, that are biomicromolecules and biomacromolecules, on the basis of their molecular weight. On the basis of their molecular weight, we can subdivide the biomolecules into two categories, that are biomicromolecules and biomacromolecules. So how we can divide these two categories, biomicromolecule and biomacromolecule? We take a living tissue. That is any leaf or any other living tissue, such as a piece of liver. We take a living tissue, we add trichloroacetic acid, We take a living tissue, we take a trichloroacetic acid and we crush this living tissue. We crush or grind this living tissue into the trichloroacetic acid, and we found a solution. Now this solution is filtered from the cheesecloth. Now we found two portions of this solution. When we filter this solution from a cheesecloth, we found two type of separates on the basis. We crushed a living tissue in a trichloroacetic acid and we filter this solution. So when we filter, we found acid, insoluble pool, that we also called as acid retentant pool, and which is separated on the cheesecloth, and another portion is acid soluble pool. We also call this pool filtrate. Now we found two types of separation pool, that is acid insoluble pool which is above the cheesecloth, and acid soluble pool which is mixed in the acid and lie in the filtrate category. Now in acid insoluble pool there are macromolecule or biomacromolecules are lies that are protein, which are protein, polysaccharides and nucleic acids. Nucleic acid example are RNA and DNA. These molecules are biomacromolecules which lies in the acid insoluble pool, and in acid soluble pool there are small molecules are present, that is amino acid or amino acid, simple monosaccharides, fatty acid, glycerols. These are lies in the acid soluble pool. So we can separate on the basis of their molecular weight. Now we should know about what is the molecular weight of acid soluble pool, means biomicromolecule and biomacromolecule. So biomacromolecules have the molecular weight of above the 1000 Dalton, and in biomicromolecules the molecular weight is 18 to 800 Dalton. Means when we talk about the biomicromolecules, the molecular weight is 18 Dalton to 800 Dalton, and when we talk about the biomacromolecules, the molecular weight is above the 1000 Dalton. And in biomicromolecules simple molecules are lies, and in biomacromolecules compound or complex molecules are found. Means example of biomicromolecules are amino acid and monosaccharide, fatty acid, glycerols, etc. And the example of biomacromolecules are polysaccharides, proteins, nucleic acid, DNA and RNA. Now an important biomolecule that is called as lipid. Lipid is an exception of this separation technique, because the molecular weight of lipid is less than 800 Dalton, but this lipid lies in the biomacromolecule. Why? What is the reason? What is the reason behind this? Because the lipid molecule is made up of two components. First one is glycerol and second one is fatty acid. This area is hydrophilic in nature. Hydrophilic in nature, and this area is hydrophobic in nature. So when this lipid molecule mixed with the water, these hydrophobic fatty acid tail make a cluster around it to remove the water from, they move from far away from the water and they make a vesicle. So when lipid molecule make a vesicle, they lies in the acid retention pool and not mixed in the acid soluble pool. So lipid is the exception of this separation technique, because lipid make vesicles and these vesicle does not pass the cheesecloth, and that's why it lies in the acid retention pool. But the molecular weight is not greater than the 8000 Dalton. but this lipid molecule lies in the acid retention pool and lies with the biomicromolecule. So this is the reason behind this. Now we should know about a very important biomolecule, that is first biomolecule which is written in the NCERT book, that is amino acid. Amino acid. This term indicate that this term is made up of two words. First one is amino and second one is acid. So we know that amino is the NH2 group and acid, normally organic acid, is COOH, acetic acid. So amino acid is made up of a chiral carbon, a amino group, and another one is acidic group. So that's why it is called as amino acid. The normal structure of the amino acid is this amino. We can also call the amino acid the substituted methane. Why we call amino acid a substituted methane? Because amino acid is made up of a chiral carbon. We say this carbon a chiral carbon because all the groups attached with this carbon are different. So in this chiral carbon, one hydrogen group is attached, one amino group is attached in N terminal, one acidic group is attached in C terminal, and other R group which is variable. Okay, this R group is variable in all the amino acid. So the normal structure of the amino acid is this. Why this is called as the substituted methane? Because as is in methane, the one carbon, 4 hydrogen atoms are attached with a one carbon. Likewise in the amino acid, 4 different types of group are attached with a one chiral carbon or asymmetric carbon. So this is the normal or common structure of the amino acid. Now there are different types of amino acid, different categories of amino acid. So we categorize amino acid on the basis of pH. Means on the basis of pH we can divide amino acid in 3 categories: acidic amino acid, basic amino acid, and neutral amino acid. So in acidic amino acid, the acidic group is more than the amino group. Okay, so that's why this is called as acidic amino acid and this act as the acid. So 2 important example of acidic amino acid is aspartate and glutamate. Okay, so these are 2 important example of acidic amino acid. The second one is basic amino acid. So basic amino acid are those amino acid in which amino group is more than the acidic group. So example, most important example that are lies in the NCERT book are histidine and arginine. Lysine is also in the basic amino acid. And neutral amino acid. Neutral amino acid are all the amino acid in which the amino group and acidic group is equal in the number. So that's why they are called as neutral amino acid. So neutral amino acid is serine, glycine, and many more. Serine, glycine, and many more amino acid are neutral amino acid. So these are the types of amino acid on the basis of pH. Second type of amino acid, we can subcategorize or we can subdivide amino acid on the basis of their availability inside the body or we take from the diet. So there are 2 types of amino acid. First one is essential amino acid and second one is non-essential amino acid. So essential amino acid are those amino acid which we take from our diet outside from the body. They are not produced or not synthesized inside the body. So essential amino acid are those amino acid which cannot produce or cannot synthesize inside the body, and there are 10 types of amino acid that are essential. So we can learn these amino acid by a mnemonics, that is try this VIP mol. Tri means tryptophan, histidine, valine, isoleucine, proline, methionine, arginine, leucine and lysine. These are the some essential amino acid. So the essential amino acid are of 10 type, and non-essential amino acid are those amino acid which synthesize inside our body, and we cannot take these amino acid from the diet. These amino acid are also have 10 types. Okay, so in our body there are 20 types of amino acids are present. In our body there are 20 types of amino acid are present, in which 10 type are essential amino acid that we take from our diet or with our food, and non-essential amino acid are those amino acid which we cannot take from our diet. These amino acid are synthesized inside our body. So this is the second categorization of amino acid on the basis of their availability inside the body or we take from our diet. Now the third one categorization of the amino acid are proteinaceous amino acid and non-proteinaceous amino acid. So proteinaceous amino acid are those amino acid which takes part in the protein formation, and non-proteinaceous amino acid are those amino acid which cannot take part in the protein formation inside our body. There are many types of another amino acid that does not take part apart from the 20 amino acid. This 20 amino acid takes part in the protein formation inside our body. So that's why we called these 20 types of amino acid proteinaceous amino acid, and another type of amino acid are not non-proteinaceous amino acid. Now an important topic of amino acid is deuterion. Okay, so deuterion is the important subtopic of the amino acid. We should know about what is the deuterion, what is deuterion is. So when amino acid is charged, both the group are charged. When both the group are charged, this stage of the amino acid is called as deuterion. Means this hydrogen atom moves to the amino group, and acetic acid is negatively charged, and acetic group is negatively charged and amino group is positively charged. So this stage is called as deuterion. This ion is found in the pH of approximate 7 in neutral pH. In neutral pH, this deuterion is formed. This form of amino acid, that is deuterion, is formed in the neutral pH. Now we know about the aromatic amino acid. So aromatic amino acid are those amino acid in which in the R group of amino acid. In aromatic amino acid, the R group is attached with the aromatic group. So these are called as aromatic amino acid. There are three important aromatic amino acid are. First one is phenyl alanine. Second one is tryptophan, and third one is tyrosine. So these are the three important aromatic amino acid. In these aromatic amino acid, the R group is attached with a aromatic This R group is attached with the aromatic molecule, so this is called as aromatic amino acid. Okay, now we know about the sulfur containing amino acid. Sulfur containing. So sulfur containing, there are two important amino acid are that are given in the NCERT book. First one is cysteine and second one is methionine. So these are the sulfur containing amino acid. So this is all about the biomolecule and amino acid. We revise the content. So biomolecules are those organic molecules that are found inside any living tissue, and we subdivided these biomolecule in the two categories. First one is biomicromolecule and second one is biomacromolecule. In the biomicromolecule, the molecular weight of these molecules are less than 800 Dalton, 18 to 800 Dalton, and when we talk about the biomacromolecule, the molecular weight is above the or 1000 Dalton and above. So this is all about the biomolecules. And amino acid are those biomolecule that are lies in the biomicromolecule because they are found in the acid insoluble pool. Their size is short and their molecular weight is less than 800 Dalton. There are different categories of amino acid, different types of amino acid. Acidic amino acid, that is glutamate, aspartate, glutamic acid, aspartic acid, basic amino acid, lysine, arginine, histidine, and neutral amino acid, lysine, serine. There and another one is proteinaceous and non-proteinaceous, essential one and non-essential one. So essential are 10 types, non-essential are 10 types. We can learn essential tryptophan, tyrosine, and threonine, histidine, valine, isoleucine, proline, lysine, leucine. There are the essential amino acid that cannot form inside our body. We take from the outside, from the diet, and essential amino acid are non-essential amino acid are formed inside our body. We cannot take these amino acid from the outside of the body. Sulfur containing amino acid are cysteine and methionine, and aromatic amino acid are those contain aromatic group in their R group are tryptophan, tyrosine, and phenylalanine. So this is all about the biomolecules and amino acid. So this is all about for today, and I think that topic is all that clear, and this is all about the topic. Thank you.